Welcome to the Biotech Support Group Product Knowledge Series on Cleanascite, Lipid Removal and Clarification Reagent. Cleanascite from Biotech Support Group selectively removes lipids, cell debris, lipoproteins, floating fats, impurities from cohn paste, transgenic milk, egg yolk, and biological samples for pretreatment of samples prior to purification. It has a high binding capacity for lipids with minimal cross-reactivity with proteins. Serum or tissue sample, upon adding Cleanascite, centrifuging, filtering, has fats, lipids, and cell debris removed, and supernatant contains clarified delipidated sample ready for further downstream analysis. Cleanascite effectively replaces chlorinated, fluorinated hydrocarbons, example freons, and it is environmentally friendly. It helps purify antibodies, recombinant proteins, nucleic acids, proteoglycans. It is also ideal for clarifying ascites, serum, cell and tissue culture, bile and organ homogenates. It clarifies saliva and fecal components. Cleanascite clarifies and purifies antibodies, proteins, nucleic acids, proteoglycans, and other macromolecules. This experiment shows egg yolk after and before treatment with Cleanascite. Let's review application of Cleanascite for bile research. Authors Farina et al. Published an article in the journal Proteome Research titled Proteomic Analysis of Human Bile from Malignant Biliary Stenosis Induced by Pancreatic Cancer, and cited Cleanascite. Using Cleanascite, scientists isolated and identified hydrophobic polypeptides in human bile and subsequently performed specialized reverse phase chromatography and gel filtration and Maldi-Toff mass spectrometry to identify a small subset of 5 proteins. Biofluid was obtained by endoscopic retrograde cholangiopancreatography from a patient with cholangiocarcinoma. Unfractionated biofluid was centrifuged and partially cleared supernatant was then mixed with 250 UL of Cleanascite followed by rotation, centrifugation, clearing away of the formed lipid micelles. Authors Chen et al. Published an article in the Journal of Hepatobiliary and Pancreatic Diseases International titled Two-Dimensional Electrophoresis for Comparative Proteomic Analysis of Human Bile, which cites Cleanascite from Biotech Support Group. A reliable method for the general cleanup of bile fluid samples, which is suitable for 2DE, by which scientists developed 2D, used bile fluid samples obtained during surgical drainage procedures and implemented Cleanascite from Biotech Support Group to remove debris, nucleic acid, and mucins followed by rotation for 1 hour. Salts, lipids, nucleic acids, and other contaminants are bound in bile fluid, dramatically affecting both reproducibility and resolution of 2DE. The objectives of the study were to establish reliable sample preparation method and 2DE options suitable for comparative proteomic analysis of bile fluid. Authors Guerrero et al. Published an article in the Journal of Chromatography A titled Contribution of Solid Phase Hexapeptide Ligand Libraries to the Repertoire of Human Bile Proteins, which cited Cleanascite. Scientists use immobilized peptide ligand libraries to concentrate dilute bile. For the detection of low abundance proteins from bile, Cleanascite was used to clarify bile. The reagent is a solid phase non-ionic adsorbent supplied as a suspension in saline ready for use. Pretreatment with hexapeptide ligand libraries opens up new perspectives in the discovery of biomarkers in human bile. Authors Chen et al. Published an article in the Journal of Hepatobiliary Surgery titled Establishment and Preliminary Analysis of a 2D Human Biliary Map. Surgical drainage of biofluid samples from patients with cholangiocarcinoma and cholelithiasis was collected, sonicated, and centrifuged to remove debris, nucleic acid, and mucins as a preliminary separation method. Cleanascite was used for sample delipidation, allowing for proper sample preparation process suitable for two-dimensional electrophoresis of biofluid. By doing so, biofluid analysis and identification of biomarkers by 2D biliary maps are visualized. Authors Christiansen et al. Published an article in the journal Molecular and Cellular Proteomics titled A Proteomic Analysis of Human Bile, which cites Cleanascite. Scientists discovered large amounts of lipids, bile salts, and biofluid obtained by ERCP from patients with cholangiocarcinoma. To identify proteins and bile fractions, researchers used Cleanascite to precipitate lipids from unfractionated bile, followed by one-dimensional gel electrophoresis, lectin affinity chromatography, and liquid chromatography tandem mass spectrometry. Cleanascite successfully precipitated lipids, fat droplets, cell debris, and mucinous impurities. 3 kDa size exclusion filtration steps subsequently removed salts. Monoclonal antibody sample preparation and membrane chromatographic column life extension is an application of Cleanascite. Cleanascite is an ideal sample clarification and purification reagent for physiochemical fractionation, antibody purification, class-specific affinity purification of antibodies, antigen-specific affinity purification of antibodies. It is compatible with large-scale antibody purification protocols. During the development process of anti-immunoglobulin E antibodies, Cleanascite is required because it is a solid-phase, non-ionic adsorbent, and this specialty technology makes it great for initial purification. It complements other purification biotechniques such as gel filtration, dialysis, ammonium sulfate precipitation or ion exchange chromatography, or hydrophobic interaction. Moreover, Cleanascite is the right choice for experiments in which affinity chromatography cannot be implemented or if more purity is needed. These are references on Cleanascite, which are on biotechsupportgroup.com. Please order Cleanascite from biotechsupportgroup.com. Thank you for tuning in to the Biotech Support Group Product Knowledge Series on Cleanascite. For more information, please visit www.biotechsupportgroup.com. Thank you.